Hey everyone, welcome back to Office Hours Unplugged, where we chat with the most prominent researchers and PIs here in McMaster. Today we bring with us to the studio Dr. Deborah Sloboda. Could you please introduce yourself to the audience and let them know a little bit about yourself?
Yeah, sure. Thanks for having me. My name's Deb Sloboda. I'm a professor in the Department of Biochemistry and Biomedical Sciences, and I'm also the Associate Chair of Research for that department, and I'm a Canada Research Chair in Early Origins of Health and Disease Risk.
Great. So could you tell us a little bit about how your journey, your academic journey, so to speak, started?
Sure. It started much the same as everybody else's. So went to school, decided that I actually didn't know what I wanted to do in high school. I really sucked at math. When we went to high school, we still had grade 13. So grade 13 math was beyond me, calculus. And so I actually started university at the University of Guelph in first year in a Bachelor of Arts program, taking French.
Wow.
I was going to be a French teacher, and with psych, and took a couple of courses because I really, really loved biology. Took a couple of biology courses, and just thought, this is ridiculous. Like, what am I doing? Gonna be a French teacher. That's not me. Transferred, and then I had to actually take a whole bunch of equivalency courses because I didn't have them to get in. So spent like the first semester retaking high school physics and chemistry, and that was unpleasant. But then, started in the human bio program at the University of Guelph. I still did a French major. That's where I started, and I took really cool courses. And in fourth year, I did a thesis with somebody at the Ontario Vet College in embryology, and thought, this is cool. I want to do more of this, and that's where it started.
And then, what did you do in grad school? Did you continue down the same sort of vein of research?
Yeah, sort of. After fourth year, I took a year off because I needed to earn some money. So I oddly sold futons and tie-dyed sheets, as you do when you live in Guelph. And then I started a master's at Western University in actually the Department of Kinesiology.
Wow.
Looking.
It's a departure.
Yeah, well, sort of. I found somebody who was interested in looking at pregnancy exercise during pregnancy.
Oh.
So I looked at the impact of exercise during pregnancy on maternal glucose metabolism. And during my master's, I had to take a bunch of courses, and then I took a course from somebody who was teaching basically prenatal development and placental function. And that individual ended up moving to U of T as a prof. And so that's where I went to do my PhD with him at U of T in the Department of Physiology. And that's where I ended up doing fetal physiology work as a PhD. Yeah.
And were there any challenging parts of your journey? I know, you mentioned, you had to take a year off, then you had to go back to doing your master's, and then I assume that was a lot of work on you, and it must have been very intensive. So was there any particular points that you found that was, really challenging or really, I guess, helped you develop as a researcher and as a person?
Yeah. The undergrad was hard. I don't know why I decided that I had to do a double major in two different faculties. That was wild and wacky. I actually, looking back, it was the greatest thing I ever did, because taking a French major means that you have to write a lot of essays and read a lot of books, albeit in French. But that really got me going in terms of understanding how to form a story, and I think that's probably the most important thing that we do in research and science, is tell a story. just happens to be a story about, biological function in my case. So that was hard. The second thing that was hard, the master's was hard. There was a big transition there. And actually the PhD was awesome. It was super, super hard, but actually one of the best periods in my life in terms of the research, because it was like really exploratory. It took a long time, but whatever, 5.5 years to do a PhD. Had a great time, met amazing people, traveled a lot. So yeah, every stage has its challenges,
Right.
but you just have to keep thinking to yourself, what's the outcome? What are the good things, about this journey?
Well, I don't know if you talked about this, but what drove you towards the study of embryology and placental metabolism? Maternal metabolism. What drove you to the whole area of research?
Well, I took a few courses in early development during my undergrad, and there was one course, an embryology course, actually a teratology course, which is the study of molecules that result in congenital abnormalities or death really early in the embryo. And I just found it fascinating, like how the heck do all these cells come together to make like a living organism? There's just so many possibilities that things will go wrong, and yet most of the time, well, actually, what we see at the end of a pregnancy is the result of 1 in 4 being lost. But anyway, so most of the time they actually are lost. Yeah. But the fact that you can form this like perfect organism at the end and everything works, and it fascinated me that the cells knew where to go and how to form a spinal cord, and right, it's so cool.
And in your journey to become like a PI, did you always know that you kind of wanted to open up your own lab? And did you maybe model it after, you mentioned that you followed a PI to UFT to do your PhD. Was he kind of the role model that inspired you to become a PI yourself?
Yeah. I think once, in fact, I'd like to say he did. He definitely got me to where I am, for sure. But I don't think he wasn't the switch. Mm-hmm. I think it was my undergrad thesis supervisor who was like, he's now retired, Ken Fisher at the University of Guelph. He was like an amazing lecturer. Everything that he described about embryo development, you could see that he found equally fascinating. Mm-hmm. And one of the things that he told me during my undergrad was that the best work that you'll ever do during your life as a scientist is when you are sitting at your desk with your feet up on the desk and just sitting there thinking. Don't lose sight of that. And I was like, well, at the time I was like, what's he talking about? Right? I got a lot of work to do, right? But I think about that a lot, that we never, ever carve out time to think. And we actually, just before we started recording, had a conversation about how writing is thinking. Mm-hmm. And, the use of AI to help you write is great, except you're losing thinking. Mm-hmm. You're not actually thinking, and without thinking, you're not learning. So anyway, I think it might have been him, actually.
I mean, yeah, like just kind of following up what you said, I feel like writing is all about having like meaning, right? You have to be meaningful with your writing, especially. I mean, when you do research, I guess in my opinion, like you kind of have to think about what's the point of research, right? Why are we trying to progress research, right? You have to think about it's all for human development, right? So we understand more about human growth, especially in your case, you're doing prenatal development, placental development. That's incredibly important to kind of understand how humans develop and maybe even target diseases, before birth, right? Mm-hmm. So I think kind of going back to writing and research, it's all about meaning, right? Mm-hmm. So I don't know if you agree with that, but
I do agree with that.
That's kind of what I think.
Absolutely. And without meaning, the work becomes pointless and mundane, right? Yeah. So that's why most PIs are really passionate, and most of us work far too many hours, but we actually don't. some of it's work. The fun stuff I don't consider work. So if I go on a holiday and I get to print off like 3 papers that I really want to read, like that's awesome because I'm interested. I'm interested, right? So, yeah.
And do you think what your PI was referring to is maybe the phenomenon of people getting way too invested in like the day-to-day research that they don't really zoom out and look at the big picture and why they're doing what they do?
Yeah.
Is that kind of what you were?
Yeah. Unfortunately, we're, for many reasons, we have to do this, but we are target and goal focused, outcome focused, right? So getting things done, ticking the boxes. I got that done. That's done now. That's great. Where's the next thing? How many more papers? How many more grants? How many more exams? What have I got done?
Right.
But sometimes we forget about the journey of getting there. And the process of getting there is also fun.
Right.
You just have to let go a little bit of the timeline. It's not a sprint. Right. I'm never going to answer all the questions that I want to answer. Literally never. So once you kind of decide as a scientist and a researcher, whatever area you're in, it doesn't have to be academia, but once you realize that, you're like, okay, I kind of like what I'm doing. I'm going to enjoy what I'm doing instead of what I'm completing. Of course, we have to complete things. I mean, that's life, right? I'm not going to get paid. You're not going to graduate, right?
Yeah.
So you have to complete things, but, don't make it the sole vision.
And was that like the vision for your lab? You were really passionate about, your research with pregnancies and then development, like prenatal development. So was that the vision for your lab, doing research that was meaningful to you and then research that you thought was, fun and, something I guess you couldn't really consider work, but, kind of work as well. So.
Yeah. So, I mean, it's evolved, of course, over the years, but initially it was the thing that I was excited to learn about. Part of it is also the fact that we understand very little about biological women's bodies. We don't. They've really been understudied, undervalued since Aristotle, I think, who, termed a lot of things that we still use today. Like, did you know that do you guys know the origin of the word hysterectomy? Do you know what a hysterectomy is?
No clue.
A hysterectomy is the removal of a uterus for a reason. Could be cancerous. It could be whatever, for whatever reason. So why is it called a hysterectomy and not a uterectomy?
I don't know. Is it because he's a historical figure? So.
No, because the uterus was a source of hysteria, of women's hysteria.
I see.
And they called them hysterical because of something
that
emanated from the uterus.
I did not know that.
That's crazy, right? So that's just a
see.
really good example of why I feel quite dedicated to making sure that we understand not just disease, because we're just not a bunch of diseases walking around, right? But to understand why things work the way they do, and how we are all individually different from each other, and why drugs that you take may or may not work or work differently in me.
And just kind of zooming out, after you did your PhD, did you do any sort of postdoctoral training before you went on to become a PI, or what was the process like?
Yeah. So I did. So let's see. I went Guelph, then Western, and then U of T. And during my time at U of T, I had some challenges with my experiments. I used pregnant sheep as a model for understanding pregnancy, which is a normal thing, which we can talk about later, but I'm not going to get into right now. But it's a standard model. And for the first 13 to 14 months of my PhD, nothing worked. And I was losing every animal I ever worked on. And my PI knew of another person in Australia that was doing similar but not quite the same work that we were doing, and it was working quite well. They have quite a robust breed of sheep in Australia called the Merino. You would know this, Merino wool, for example.
Oh, yeah. Okay. That's ringing a bell.
Yeah. So he said, okay, I think you should just go to Australia for 6 weeks and do some experiments there. And I was like, what? Like live in Australia for 6 weeks? What do you mean? And he said, yep. So I was new to animal work because I hadn't done. I did exercise in pregnancy in women.
Right.
I was new to animal work already, and now he's carting me off to some far land.
Laying down under.
Yeah, and I ended up in Perth, Australia. And for those of you who don't know, most people have heard of Melbourne and Sydney.
Yeah.
It's like from here to Vancouver away.
Oh wow.
So it's really, really, really in the middle of nowhere. It was very beautiful. So that was a little bit scary, but actually it ended up being an amazing thing because I went down 2 or 3 times during my PhD, which is incredibly privileged to be able to be sent to Australia and live in Australia. Collect all my tissues and then ship them back. And then I was offered a postdoc once I was done. So I went back to Perth, Australia, with my husband and the fetus that I was carrying at the time because I became pregnant in the last year of my PhD and did a 5-year postdoc in Australia, where I set up some labs in their maternity care hospital. They didn't have wet labs there. They only had path labs. It's quite an old building. It was built in the 1930s. Super cool building. But you can imagine it was like not set up for, the stuff that I was doing. And then after that, I had 2 kids there. And then after that, I had decided it was time to go back to Canada, and then somebody in Auckland, New Zealand, found out that I was leaving Perth and offered me a job there. So I did kind of another postdoc at the University of Auckland.
It's amazing.
And then I spent 5 years there, and then moved back here and came back to
Must have been whiplash.
Yeah, I miss New Zealand a lot, actually. Yeah.
So how was like the experience? I know you had experience researching in Canada and then U of T, and then you did your master's in Western. But how is the research in Australia? I know, I guess different countries have different perspectives, different techniques, and for research, I think that's really valuable. So I'm kind of curious, like, how did you find, I guess, the contrast between how research is done in Canada versus Australia?
Yeah, the research and the experiments themselves were the same. We're all kind of the same. The major difference is in Australia and New Zealand. It's a little bit more, the funding scheme is a little bit more like it is in the United States, where you actually have to apply for your own salary. So when you apply for a grant in many places in the United States and in Australia and New Zealand, you actually put yourself on as staff and get your salary. So which is great if you get a grant, but not so great if you don't. So it's actually really, really stressful. You're having to supply yourself with salary and supply the operating funds and also fund everybody else that's working with you. So that's different. It's far, far away. Where I worked in Perth is really far. It was quite isolated. There wasn't a lot of contact, and to go to the next nearest city was a 4.5-hour flight.
Wow.
Jeez.
So the nearest city that does research is what I mean. There were plenty of cities we could drive to that made wine, right? But that wasn't gonna introduce me to other researchers. So that felt very isolating. It was time to go, right, after a while. And then New Zealand was great. New Zealand felt a little bit busier in terms of science and research because I ended up going to one of the most important institutes for fetal growth and development. It was set up in the early 50s. And some of the biggest people in fetal growth and development worked there. So that was really wonderful. So I got to meet the people whose papers I was reading, like that were published in 1968. He was still there going to the seminars. And, I think Sir Graham was maybe 85 and sleeping during most of the seminar, but always ask the best question. Right? One of those where you think, how did that happen? So I had some really great experiences there. And I think because those places are isolated, they tend to have a different sort of culture in and of themselves, like a community culture. But eventually, you know, like 4.5 million people live in New Zealand, and it felt small. So we wanted to come home. We wanted to be Canadian again. Missed Canada a lot.
And kind of shifting gears towards your lab, because one of the most unique parts of your lab is that you're really involved in community services. I know you have something called the Art of Creation, and I think that's so interesting. I mean, we don't really see that many labs that kind of get involved in the community. So we're kind of curious, like, how did that start? Where did the idea kind of originate from?
Sure.
And we just want to learn more about it. It's very interesting.
Yeah. So for your listeners, the Art of Creation is an arts-based knowledge translation program where we basically storytell through the use of art about early development. And I did this in partnership with the Art Gallery of Hamilton and with Sarah Dickinson at the Art Gallery of Hamilton. Look, you know, I've been doing this and fascinated by this since, I'm going to date myself here, let's see, 1992, before many people were born. 1992 is when I took that embryology course, 91 actually. And, you know, here I am in 2000s, in the 2020. Nobody knows what I'm talking about when I say, did you know that your early life environment actually shapes your disease risk later in life? They're like, oh, yeah, my genetics. Oh, but we don't have any cancer, let's say, in our family, or we don't have any genetic abnormalities. And I'm like, I'm not talking about genetic abnormalities. Like, totally just the environment within which, you know, the oocyte and the sperm develop and how the fetus grows and develops. That's it. Nothing to do with genetics. Nobody knows what I'm talking about. And I kind of became frustrated with it. I became frustrated with the fact that pregnancy is this soul journey that one individual goes through. And it doesn't have to be that way. You know, there's actually a partner that contributes sperm to the pregnancy that doesn't know anything about it. We don't even know. We still don't really know the process of labor and delivery because if we knew that, we could possibly prevent preterm labor, which is how a lot of babies die. And I thought, obviously, whatever I am doing, you know, I've been doing it for like 25 years. I'm obviously not doing a very good job telling this story because nobody knows what the heck I'm doing. So I just kind of went, a lot of the stuff that we look at in the lab, it looks like art. I always thought that. We did a lot of work on ovaries. Ovaries are so beautiful down the microscope, the oocyte and the follicles. It's like, well, maybe, I don't know, could we do art? Could we bring people to the lab? What could we do? And so I basically called Sarah and said, is this a crazy idea? And she said, no. I love Sarah because she never says no to an idea. She's like, yes, we could do that. Anyway, so that's how it all happened. So basically we did a whole bunch of focus groups of pregnant individuals in Hamilton. They did artwork. We talked about really sensitive and controversial topics using art. But we also brought artists from Hamilton into the lab, and they created art based only on the experiments that they witnessed and the conversations that they had. And we put it together and we made an art exhibition and we took it to the community. And we worked a lot with public health, and we work a lot with services that provide support for pregnant individuals in Hamilton. And the exhibition is touring around. It's on its way actually to Western University.
Wow.
Next month. It's going to be in the Weldon Library. If anybody comes from London, you can go and have a look at it. Then it's going to be shown at the British Science Festival in Southampton, UK.
That is...
Yeah, that's a big deal. We're still trying to figure out, how do you pay to get it over there, but we'll work that out. and it might go to Berlin, and then it's going to come back here to McMaster MMA in 2027. So
Wow, big project. Wow.
yeah, huge project.
It's the year we graduate.
Yeah, good. You
will
can come
not
by
miss
later
it.
and see it.
That's amazing.
And I don't know if you mentioned this. When did your lab start this initiative?
Oh, we wrote the grant and got funding in March 19th, 2020.
Wow.
What else happened
Oh my
in
God.
March 19th, 2020?
Yeah,
Yeah.
Well, it
So
must have been a curveball.
it was a curveball. It was. So basically, we didn't do anything
Mm.
for 2 years. And even after 2 years, I was really nervous because we know that SARS-CoV-2 was terrible for pregnant women. We didn't want to expose anybody, right? It was pretty stressful for the first year and a half of us getting together and working with community, simply because we were working with such vulnerable people, right? And they also sometimes came with their children, and I didn't want to expose children.
Yeah.
So it was tricky, but we did it. Yeah, totally. And it was a great experience. All the people that were involved really, really loved it. And the launch of the exhibition was in the Art Gallery of Hamilton in the community space where all the pregnant individuals and the artists could come in and see it. It was there for about 3 months.
Wow.
Yeah.
It must feel very gratifying to see you do your academic work in the lab, and then you actually sort of apply the main message of why you do the work that you do, and it comes to fruition like that.
Yeah, and see it on display as well. That's amazing.
Yeah. The ultimate story, right?
Yeah.
Like I said in the very beginning, I was
yeah.
interested in stories, and this was the ultimate story. I think there were two great accomplishments in that particular project. Is that number one, we brought pregnant women and people of Hamilton together that didn't have a network. They said that. They said, I didn't know anybody in Hamilton, and here I know so many people now. They formed Facebook groups. They formed, right? So we gave them that opportunity. That made me feel like, great, awesome. I can walk away, right? I did something for people that needed support in Hamilton during their pregnancy. And then the second thing was that learning that we often underestimate, what we call the lay public, right? Non-experts, because we brought artists into the lab. I did not give them a lecture. I did not, explain to them the definition, of embryology and early development. They just asked questions, took notes. They talked to my students. They talked to me. They saw experiments. They asked, why are you doing these experiments, and absorbed the information, walked away and produced these amazing pieces of art that have such meaning. And I was like, what? They get it. They totally get it. They did not need to listen to me drone on, right, in a 50-minute lecture about the process of placental vascularization to get it. And they got it. And it was super cool. And I had one particular artist. He's a rapper, LT the Monk. I don't know. Some of you might know him.
Yeah.
He's on Spotify. He came out in his lab. Maybe this is too much information. I think LT would allow me to say this. In his last session in the lab, he's like, I was like, how did it go? And he's like, great. I have to change my whole life. And I went, what do you mean? He goes, oh my God, I'm wrecking my sperm. And I was like, okay. So,
Very interesting.
right? He got it. He totally got it. Because that's it. That was the whole point.
That is really cool. The whole initiative. That is amazing.
Awesome.
I think that's a good. Honestly, I would take that as a compliment.
That's a win.
Yeah.
Yeah, it's a win.
Everyone's becoming educated. That's amazing.
Totally.
Really
and that
good.
was the whole point of the exhibition, is to get people to understand it without me, as I said, graphs, and that doesn't work. Didn't work for 20 years.
Well, kind of zooming out from that, looking more into what research your lab does. Is there any project that you're currently excited about within your lab that you would like to talk about?
Oh my God.
We're not asking you to
Too
pick
many
favorites.
Too
but-
many projects in the lab. Yeah.
Maybe to narrow it down, like, is there any do you think would be, I guess, really interesting to the public, or you think really, I guess, relevant to the public, kind of similar to the art of creation? So something research that the public might be interested in, or they can.
Sure. So I'll give a broader overview a little bit. So one of the things that we've done is not just focus on the maternal lineage. So most people, when they think about early development, they default to obviously, what the pregnant individual is eating, doing, saying, moving, right? That's all going to impact on the condition of pregnancy, which is true. And that's what we study. And we try and figure out, what happens if we change macronutrient intake or change, adiposity before and during pregnancy to look at placental blood vessel development. We do a lot of that. There's some very cool data that we have out of that. We want to understand how pregnancy changes women's bodies. We do a lot of that under normal pregnancy conditions and under conditions that might be stressful or changing nutrition. We do that too. And we embarked on some new studies like about 6 years ago to understand how the maternal gut microbiome influences adaptation to pregnancy. So we've done a lot of work on that, which is really cool. I think I would like to think that that's a pretty cool contribution because we as perinatal biologists never said to ourselves, "Oh, I think the intestine might contribute to pregnancy." Like it wasn't a thing, and now we know that it does. And why wouldn't it? In retrospect, duh. Like it's an organ in the body. Why wouldn't it actually contribute to some of the changes that you see? So we've done a bit of that. That's been pretty cool. But we also have investigated the paternal lineage because, as I said, we default to the maternal lineage simply because that's the person who is actually gestating.
Right.
But we and others have discovered or rediscovered, I suppose, have discovered it. It existed all along. That the paternal condition, not just sperm, but also the factors contained in seminal fluid, which are really important for implantation, actually can influence placental vasculature
I see.
completely separately from the maternal condition. So, and after controlling entirely for the maternal lineage, if you play around, let's say, for lack of a better word, with the paternal condition, you can actually change placental function.
Wow.
So, I'm going to say 0.1% of people in the world know that, right? So most people just think that sperm contributes DNA, and that's obviously, the epigenome and the genome, it contributes that. So that must be obviously what changes fetal development. Let's say if the father is smoking or taking drugs or whatever, it's going to damage the sperm. But the thing that most of us don't think about is how the sperm and things like cytokines in the seminal fluid actually influence both implantation of that embryo and the growth and development of the placenta. So, and if the growth and development of the placenta is altered, you will alter fetal development completely separately from the genome and the epigenome, because the placenta fuels the fetus, right?
It's fascinating. Wow.
Yeah.
Oh my God.
So I really like that. So that's what I mean, I don't know which one I like the best. They're all good.
Yeah. That was wrong of me to ask you to pick favorites.
My lab always says, "Who's the favorite?" And I'm like, "There are no favorites."
Actually, I have more of a specific question because I always like sometimes I see these videos online when a woman goes through like pregnancy, right? Like as you know the baby develops, all the organs are kind of being smushed together. And is there any like long-term side effects of that? 'Cause, like it's like seeing it is
Being squished?
Yeah, it's like it looks so brutal and painful.
Yeah.
I'm like, is there any long-term effects? Is there
No.
any way we can alleviate that? It's
No.
like it's very
Yeah.
I don't know. It's
So being squished is normal.
Mhm.
I will tell you. It makes me think of a series of experiments were done in 1938. And one of the things about pregnancy, there's this concept called maternal constraint. So if you can imagine, and people say this, right? Let's take a male basketball star, right? Who happens to have a partner who's 4'2", and they have children. And everybody says, "Oh my God, how does that happen?" Like the genetic potential of that baby, right? Whose paternal contribution is 7'5". I don't know. I'm making it up. Obviously, right? That's crazy. How does that happen? Well, it happens because of this concept of maternal constraint. And there's epigenetic control of growth and development. So a lot of the maternal alleles actually prevent fetal growth, because if fetal growth isn't controlled, then the mother will die during birth as well as the baby, obviously. And a lot of paternal alleles drive fetal growth, right? Because the father wants the biggest, right? Because let's face it, like in the world of animal ecology around the world, we are interested in passing on genetic material. So the paternal lineage is interested in large organisms. So the theory is that this maternal constraint has evolved over time to prevent the fetus from outgrowing its environment.
Nice.
So there's all sorts of constraints. That's one of the constraints. There's lots of other constraints. Blood flow, blah, 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 and the way the placenta develops. But that's a cool concept. So what they did in 1938 was they took they took some horses, they took ponies and thoroughbreds, and they crossed them. So they made embryos, and then they crossed them and said, well, what would happen if I took, a basketball player? But, how does this happen, right? And what they found was that if you look at the embryos that were implanted into thoroughbreds that came from ponies, so small and to big, they grew to the size of the thoroughbred. It was just like one big honking pony, right? Not a pony, but a big pony. But the thoroughbred was somewhat growth restricted. So some of the environment, it actually does end up being not amazing. But in humans, that's not been shown, obviously. It doesn't result in anything wrong at all. Like, it's, it works. It works just fine because of this concept of maternal constraint. So, being squished is all okay. Having said that, the evolution of C-section has changed the way fetuses have grown, because evolutionarily our head size was dictated by the ability of the biological woman to be able to deliver the baby, right? Because of the pelvis. But now that we have C-sections, because C-sections happen sometimes if the baby is too large, or if the head is too big. So what happens if those people continue to have babies?
Over time, that's gonna
over time
that maternal constraint would probably be kind of alleviated.
Exactly. Because part of the constraint is everything, right? The size of the pelvis, the growth factors, the epigenetic, blah, blah, blah.
That is pretty fascinating.
Yeah, so it's interesting how you can actually think about it in terms of evolving changes. And that's why I find just regular old pregnancy, right? Nothing complicated. Fascinating, right?
Yeah, it is fascinating.
So many different factors, the paternal, maternal, then the actual development of the baby. during, I don't know, so many weeks, so many months, and then you kind of see the progression, and like
Yeah.
one phase of pregnancy is different from another phase of pregnancy.
You
It's
got
so fascinating,
Yeah.
so complex.
Yeah. I
So-
picked a super complicated area, didn't I?
Yes.
I should have just picked one protein.
That's what labs do.
How does
so-
it fold? Yeah.
Just one more question, because I think we're running
Yeah.
out of time a little bit.
Yeah.
I guess this might be hard, but I'm just very curious. Out of all the accomplishments as a researcher, and not as a person, because then we'll be getting into other stuff, but as a researcher, is there an accomplishment you are most proud of?
Probably the community-based
The
stuff.
community one, yeah.
Yeah. It means that all the work and excitement and everything that I did in the lab actually gets out there.
Right.
Because I'm not here to make really healthy sheep or mice or rats. I want to make really healthy humans and support those people that are pregnant.
Mhm.
So I'd say that.
Hmm.
Yeah.
It's very nice.
Can I
Well,
add to that actually?
Yeah,
Yeah.
go ahead. Yeah.
Go for it.
So that, and also the students that I've worked with. I'm super privileged to work with, and have worked with many people, and honored that they chose my lab to work in and graduate from. So those are the two things. I'll say two.
Amazing.
I feel like most of the people we talk to tend to cite that as one of their main accomplishments, is seeing their work translate into real people,
Yeah.
actually having an impact on real people, essentially,
Yeah.
whether it's people in their lab graduating, moving
Yeah.
on, or it's initiatives like the one that you made. I feel like that's kind of what sticks to people the most,
Yeah.
I
Yeah.
would say.
It's like
Yeah.
passing the torch down, you know?
Yeah.
You did all this research, now you're like, your work is like a foundation for someone else, and it's like only like the research will only keep growing and growing and growing, and eventually, I don't know, maybe we'll figure out exactly how pregnancy works. Every step will be figured out in maybe a century or two, maybe.
Maybe
But
we will. Maybe we will.
Being a little hopeful here.
Yeah,
but-
exactly.
Well, thank you so much for coming down and taking the time to talk to us, Dr. Sloboda. It's been an absolute pleasure hearing you talk about your work. And thank you to all our listeners, and we'll see you next time on Office Hours Unplugged.
Thanks.